Okay, hi everyone. So today we are reviewing for our exam 3 that's coming up. As a reminder, our exam is on Monday the 10th, so that's this coming Monday, and it is at 12 noon. So it's kind of a weird time, but that's because we're using the time that was set aside for our class based upon the final exam schedule. But recall that the exam itself is not going to be a big cumulative final. It is just our third exam for the course. So it'll be the same format, same experience that you've gotten used to from the first two exams. So in other words, all you got to do is at 12 noon on Monday, remember to log into La Lima, right? You don't have to worry about Zoom. Just log into La Lima, and the exam will appear at 12 noon. So I think up until that time, you won't see it, so don't worry about that. But at 12 noon, it should appear. You go ahead and click Begin Assessment, and you'll have 50 minutes from that point to answer the 50 questions, just like the two prior exams. All right. So you'll have a 10-minute sort of grace period, so you can log on and start your exam no later than 12:10, right? And still have the full 50 minutes before La Lima closes the exam down at 1 o'clock. All right, good. So the exam will cover everything from week 12 through week 16. So that is chapters 12 through 17. So let's go ahead and review that stuff. Oh, before we get into the review, I also see here, I wanted to remind you, you do have your one last quiz that's due this week. So quiz 13, which is on the chapter 17 stuff, that's the cancer and the immune system material. That is due tomorrow night, Thursday night at 11:55 as usual. All right, so don't forget, because that would kind of be a real downer if you actually forget to do the last quiz of the semester. All right, so look for that. All right, let's go ahead and review. As usual, there's no way I can go over every single lecture that's on the exam, but let's go ahead and touch upon some of the main kind of concepts. All right, so chapter 12 dealt with the coevolution of our innate and adaptive immune system. We've certainly seen this cartoon a lot over the course of the semester. It's a nice reminder that the two sides, our innate side and our adaptive side, all, of course, originate from the hematopoietic stem cells in our bone marrow that give rise to all the different types of cells that participate in our immune system and our components of our blood. So you'll recall that the innate side of our system was the one that we evolved first, right? So multicellular life, complex organisms, like animals, right, all have innate immune systems. But really it wasn't until the development of vertebrates that we began to also add on to that an adaptive side, right? And that came about because as bodies got more complex, more cells that make up the body, and the lifespan of organisms got longer, it became more important that the organism has an immune system that can not only come pre-equipped to deal with things that evolutionarily, right, you'd see over and over and over again. So it's worth it to come pre-programmed with an innate immune system that can readily detect and defend against common everyday things. But importantly, if you're going to live for several years, then it's almost inevitable that you're going to be exposed to things you don't come pre-programmed to defend against. And you either have to adapt to that and figure out a way to protect the body against it, or you're going to succumb and die to it. Right? So this chapter had to do with us appreciating the fact that it's not either or, right? Your full immune response is actually a combination of these innate and adaptive effector cells working together. And there are some examples good examples of where different components of the two sides can very easily be appreciated as sort of crossing over. An example of those that I think should come to mind are things like antibody production, right? So the B cells that give rise to antibodies, that's squarely part of our later evolved adaptive side of our immune system, right? The lymphocytes, B cells, T cells. But as we've learned, the antibodies that are produced by our B cells absolutely help out our our innate immune effector cells do a more do a stronger job. Things like antibodies that can coat the surfaces or be held onto the surfaces of mast cells, right, or other granulocyte cells, thereby sort of suiting up these cells and giving them more targeting ability. We also looked at cells like the NK cell or the natural killer cell, as seen here in purple, right, as sort of the one cell that kind of represents the first step towards the adaptive side, but the natural killer cell still sort of functions more like an innate effector. So we learned that natural killer cells, the reason they are sort of the first step towards an adaptive immune response is that in this case, the natural killer cells, right, aren't concerned with seeking out and destroying pathogens directly. Remember, the cells of our innate side, like macrophages and neutrophils, right, they're designed to just phagocytose or eat up things like bacteria to destroy them. But the NK cells represent the first step of the immune system's evolution to start paying attention to normal body cells that get infected by things like viruses, for example, right? Because if you can eliminate those infected cells from the body, that is a tremendous way of stopping the spread of disease within the body. So NK cells come equipped, you'll recall, with an assortment of different receptors on them. There's a big library of different receptors, that the NK cell can draw from within the genome. So a given specific NK cell will only express a handful of these receptors so that it can have those particular receptors in large quantities on its surface. Right? And therefore that particular NK cell would be better suited or more sensitive to certain types of signals than maybe another different NK cell that expresses a different assortment of receptors on its surface. Right? And the point is, because you have a big population of NK cells, then you're good. You have everything sort of covered, right? As these different NK cells can all kind of contribute in different ways. So you'll remember that an NK cell here, when it receives messages that its receptors on its surface are binding to things that are being shown to it on the surface of a cell that it's synapsing with, these receptors that it uses are combinations of certain receptors being stimulatory, activating the NK cell, encouraging it to release its cytotoxic cargo that it's carrying around in these little, in the cartoon here, little purple vesicles or granules, right? That might tell this virus infected cell to go ahead and die through apoptosis. Or the NK cell might end up having inhibitory receptors that are binding, right? That might discourage that NK cell from activating. And you'll recall a common theme here with most of these types of cell-to-cell communications in our immune system is that it's always a balance of activating signals and repressing signals. And it's whichever sort of side wins out when you have two cells pressed against each other, that will determine what happens next. So this cartoon is a nice reminder. You have 4 different NK cells over here, each of which has sort of a different assortment of receptors on its surface. So this cartoon illustrates that concept of plus and minus sort of receptors in sort of like this tug of war to determine what the NK cell will do. I did not give you a lot of different examples of NK cell receptors. I showed you a table back in our lecture. Maybe this is like, I don't know what this is, lecture 26 or so, that listed a whole bunch, 30 or so different NK cell receptors, but then we like highlighted maybe like 4 or 5 of them to use as standout examples, right? Just to get this concept across that some are stimulatory, some are inhibitory. So I expect you to at least know those highlighted ones. For example, in this cartoon on the left, this NKG2A receptor, right, this green one, is an inhibitory one. So when the NK cell has that on its surface, or this particular NK cell has this one on its surface, and it interacts with a healthy body cell that is expressing normal levels of HLA proteins, in particular HLA-E, right? Then the NK cell might use that as evidence that this cell seems to be healthy, right? And as a result, the NK cell is not going to activate against it. On the other hand, you would have stimulatory receptors like this NKG2D or 2B4, right, that in this case don't see any sort of message, right? So the overall message is the NK cell here leaves this healthy cell alone, right? Then it separates from it and it goes off and bumps into another cell. Maybe that next cell that it bumps into is this one in the middle cartoon, this virus-infected cell. And it is expressing all kinds of sort of weird proteins on its surface, like the M1C and this CD48, and that's what's picked up by these stimulatory NK cell receptors, then causing the NK cell to go ahead and kill this infected cell. Remember that most of the time, the NK cell needs to see 2 different stimulatory receptors activating in order for it to feel confident that, yeah, it should go ahead and release its cytotoxic cargo to kill the cell. Exceptions are things like if the receptor on the NK cell is this Fc gamma receptor, because Fc gamma, right, it hangs on to the stem of IgG. And if this IgG is present in your body and this IgG is binding to something like antigen on this target cell over here, then the very fact that you even have this IgG made in the first place already tells you, yeah, this antigen has already at some point been decided to be something that needs to be targeted, right? So in this case, the NK cell, all it needs is to have its Fc gamma receptors responding, and that is a high confidence signal that this target cell should be destroyed. Remember that NK cells also have receptors that detect different HLA isotypes. We already looked at one that detects HLA-E, right? But remember, we also have these highly polymorphic HLA-A and B proteins that are on the surfaces of our cells that evolve to be able to display different types of peptides. Right? So our NK cells want to be able to also detect those in order to be able to tell if a cell is healthy or not. But because they're polymorphic, it means that the NK cells have to have slightly different versions of these killer cell immunoglobulin-like receptors, or KIRs, that they use to detect polymorphic HLA types. And you'll remember that even if something like HLA-A has something like 1,500 different variations cataloged in the human population, different variations, the reality is there's really only like maybe 4 of those polymorphisms that, like 98% of the population has, right? And then the other, 1,000 or so different variations that have been found, right, are very rare. So NK cells evolved to have different KIRs that can just detect some of these more common variations. We ended our discussion about coevolution with another example of cells that look like they're from the adaptive side, but actually function more as innate effectors. And those are our sort of funny gamma delta T cells. You'll remember that gamma delta T cells are made in the thymus as part of this normal process of a progenitor cell that came from the bone marrow, made its way to the thymus, was convinced through peer pressure of the thymic epithelial cells, convincing it to try to commit to becoming a T cell. And then you go through this process of VDJ recombination, right? That can either lead to alpha beta T cells being made, which are the more sort of commonly thought of T cells when we talk about our immune system, right? Most physicians, doctors, if you mention T cell, they would think about alpha beta T cells, right? But now we know as immunologists, we also have the possibility of making these gamma delta T cells, whose T cell receptors have far less variation in shapes compared to alpha beta T cells. And these shapes are more designed to detect common sort of funny signals or bad signals presented by the cells of our tissues of our body when cells are damaged or old and need to be recycled or potentially diseased, right? So the gamma delta T cells play a much more innate-like role in being just sort of maintainers of normal tissue sort of function and health, instead of always looking out for novel pathogens like their alpha beta T cell brothers and sisters do, right? All right, chapter 13, we moved on to discussing immunity in the context of different diseases. We learned that we are in this constant sort of battle with pathogens co-evolving with us, viruses, bacteria that have been with us, right, from the beginning of the human species and have been constantly sort of mutating, evolving to find ways of getting around our immune system. We learned some pretty neat, clever examples of these types of strategies. For example, certain bacteria express toxic proteins that might function as things like superantigens that interfere with the ability of our immune system to properly work. The superantigen example is particularly devious, right? Because you'll recall what it does is it actually causes non-specific T cell receptors to bind to MHCs, even though they aren't the right shape to be the right combination of T cell receptor binding to little peptide on this particular MHC2. These superantigens just force them together. And when I first taught this to you, I asked you, doesn't that sound kind of crazy? Because you would think the last thing that a bacteria wants to do is wake up a whole bunch of T cells, right? But the reason why this is effective is that it causes an overload of T cells activating, right? You have something crazy like 20% of all T cells that get sort of influenced by this bacterial superantigen to contact some antigen presenting cell, some peptide on MHC. Up to 20% of them will activate, right? That is a huge portion of your CD4 T cells, right? Normally, out of all the CD4 T cells in your body, if you had an infection with a pathogen, well, it's probably less than like 0.01% of them that normally would be specific enough to activate against a critical peptide, right, being presented by an antigen presenting cell. But if you have 20% of all your CD4 cells activating, then that causes so much noise that the bacteria ends up sort of sliding below this kind of smoke screen. I see one of you asked me, are these slides going to be posted on LaLima? Yes, I will post these on LaLima once we're done reviewing. All right. We also learned that certain diseases that might be hereditary that result in immunodeficiencies, right? So remember, a very important definition is what is an immunodeficiency? Again, it means there's something lacking in your immune system. A critical component is missing, right? It can either be a particular protein or it could be a whole group of cells that are missing, right? So that means you are deficient and your immune system cannot work at its 100% capacity. We said that there is some redundancy in the genes that code for different parts of our immune system. So certain receptors, if you have a weird version of it that doesn't work well, chances are you still have a pretty good immune system, right? Unless it's a supercritical receptor, right? So if you have a defect in something like your complement receptor 1, for example, or you have defects in things like your CD4 or CD8 receptors, those are pretty important, right? And your immune system would really be compromised. But a lot of times other immunodeficiencies, there's some redundancy there. However, the most sort of impactful or severe immunodeficiencies often involve loss of T cell function, right? So we learned about severe combined immunodeficiency or SCIDs. So I think this was the lecture that I asked you to just look at the YouTube video from last semester, because this was the Friday when I was involved with another presentation that I had to give on campus. So I wasn't able to do our lecture live, right? But since I had the recording from last semester, I told you guys just to go ahead and watch that, because it's essentially the same lecture. Right? So we learned about human immunodeficiency virus as a good example of immunodeficiencies that don't come about because you are born with some sort of inherited, genetic makeup, allele in a gene somewhere that causes immunodeficiency. HIV is an example of where immunodeficiency comes about when you actually are exposed to a pathogen, right? And that pathogen, as the part of its course of disease, causes the immunodeficiency. So HIV infects cells of our immune system, such as dendritic cells, macrophages, and importantly, CD4 helper T cells. And the virus binds by taking advantage of the CD4 receptor on the surface of T cells, and also the CCR5 immune receptor on these cells. And because it eventually leads to the destruction of your entire population of CD4 helper cells, right? This leads to a loss of your T cell arm of your adaptive immune system. All right. Chapter 14, we moved on to things that I think are perhaps more interesting on an everyday kind of level of discussion, things like allergies. And we looked at the role of IgEs in allergy. So you'll recall that allergies are based upon these, what we call hypersensitivity models, right? And we have 4 different hypersensitivity models that can explain the entire range of allergies that people experience. You'll remember that types 1, 2, and 3 all involve immunoglobulins, right? Involve antibodies, either IgE that's made that ends up being suited up by a something like a mast cell that has Fc epsilon receptor, these little green things that hold on to the stems of IgE, causing things like mast cells to then release histamines and other types of inflammatory irritating compounds that cause things like sneezing, itchy eyes, itchy throat, constriction of smooth muscle. Right? And that can lead to very severe life-threatening allergic reactions. Right? Again, because mast cells are all throughout the mucosal layers of things like our respiratory tract, right? and our GI tract. So if all of a sudden you have all of these mast cells that have these IgEs on them, that, remember, your immune system improperly made, right, as part of making a mistake at some point and selecting for B cells that make this particular immunoglobulin, right? That was a mistake that your adaptive immune response made. Right? And now, as a result, when you're exposed to this actually very benign, non-threatening antigen, like pollen or pet dander or something in food like peanut and nut oils, right? You have this dramatic release of histamines by these mast cells, and therefore you end up with things that can be life-threatening, right? Like constriction of the airways or going into shock as your circulatory system, right, loses blood pressure dramatically. Type 2 and type 3 involve IgGs. Right? And in this case, these IgGs might function sort of how we normally think they function. Oh, so keep in mind, in this review slide, what I've done here is this top sort of row going across, I've labeled them as normal function, right? In other words, that's how these different components normally should be functioning to protect you against things like parasites, right? So normally over here, the IgE that's suited up on mast cells help these mast cells target something like a parasite, right? In type 2 and type 3, the IgGs normally should be doing what? They should be doing something like helping out macrophages have an easier time eating bacteria, right? So these blue IgGs are specific for the bacteria, and they make the bacteria sticky so that the macrophages have an easier time eating them, right? And also this IgG, these blue antibodies, can also recruit complement proteins to help out as well. Right? Also, pathogenic antigens like this molecule over here can be bound up by IgGs to form immune complexes. So thereby neutralizing these types of potential threatening molecules, maybe they're toxins, poisons, whatever, right? And forming these complexes of antibodies that then limit the ability of these toxins or whatever from actually getting into the cells. And instead, now you can have your other immune cells come and maybe phagocytose or eat this immune complex and destroy the threat. Right? In hypersensitivity, then, during allergies, type 2 is when these IgGs improperly bind to foreign-looking or different-looking shapes that are presented on the surfaces of your cells. So oftentimes we said that this can happen if you have allergies against certain medications, like penicillin will cause different sort of modified antigens to appear on the surfaces of your cells. And again, your body at some point made the mistake of thinking that this is actually a threat, right? And so that's why you have these IgGs as part of your immune memory. And that's what now causes your allergic reaction to penicillin. Type 3 happens when you have things like therapeutic antibodies that are given to an individual. And then you have antibodies reacting against other antibodies, causing over time a buildup of these immune complexes that your tissues of your body are having a harder and harder time clearing. Right? And then lastly, type 4 hypersensitivities have to do with T cells making the mistake. Right? The classic example are things like cheap jewelry that are made from like nickel, for example, because nickel ions are notorious for integrating with or binding to different amino acids. So now when cells make proteins with those amino acids, the shapes are kind of funny because they have these nickel ions on them. And so again, right, it still requires that your adaptive system makes a mistake and actually thinks that this is some sort of threat. Right? So the T cells then go and cause damage to the tissues that are expressing these. Right? All right. So for allergies, they follow type 1, 2, 3, and 4. Right? For tissue rejection, because that's the next thing that we moved on to, right? They're still considered hypersensitivity reactions, but they typically have to do with type 2 reactions, which have to do with you already having IgGs against ABO blood types that are different than yours, right? And this can happen then very quickly, again, because normally we're talking about mismatched blood transfusion. So a lot of blood is being brought in, right? So you have a strong reaction that can happen right away, very quickly. So we say that's hyperacute due to ABO mismatches. The type 3 and type 4 are chronic tissue rejection, having not to do with ABO blood type mismatching, but having to do with the wrong HLA profile, right? Because HLA proteins are so polymorphic, your assortment of HLA alleles that you inherited from mom and dad might be very different than the assortment that your friend inherited from her mom or dad, or her mom and dad, right? And as a result, the HLA proteins, the MHC platforms that are presented by your body's cells are different shapes. So if you transplant something like an organ, like a liver, from one body to the next, right, the patient's immune system will reject it because it will see that there's something funny and different. Right? And that can either happen very quickly if T cells are involved directly. So within a few days, the organ might be rejected, or it could happen over many years. Right? But what I taught you is that eventually, right, our immune system, because it's designed so well and fine-tuned so well to be powerful in detecting and rejecting foreign antigens, right, that even in the case of a tissue transplant where you want the kidney, for example, to survive and function properly for the rest of the person's life, right? It just simply won't happen, right? Because the person's immune system will eventually reject it. And that's why I taught you that patients who undergo organ transplants, they have to live the rest of their life on strong immunosuppressive drugs, right? To try to keep this rejection under control. And the other thing I wanted to remind you is tissue rejection very rarely, if at all, would involve a type 1 IgE mediated hypersensitivity. Why? Because, right, there's no reason why your body would have previously been exposed to, let's say, this other person's blood or this other person's tissue, like a kidney, in order to even make a mistake where your body decides to have memory IgEs around, right? So that's why with tissue transplantation, we typically only see type 2, 3, and type 4. All right, so spend some time with this summary slide. It's a lot of stuff on it, but I think it's put together in a way that hopefully will help you piece this all together. Right. We said that common allergens oftentimes resemble parasitic antigens. So an example that we used is having to do with things like food, for example. So we looked at a case where, curiously, the papaya here, right, it produces this particular enzyme, this papain, which is normally part of the fruit ripening, right? So this enzyme breaks down proteins to make the green papaya nice and soft, right? And that's why the fruit ripens that way. But unfortunately, the structure of the papain looks a lot like, for example, this protease cathepsin that's made by certain parasitic worms, like this blood fluke, right? And this blood fluke makes this because, hey, guess what? When it's in your body invading your tissues, it wants to digest and break down your tissues, right? So an immune system can make a mistake, right? And one day, for some reason, if it's exposed to this papain, it might actually think that this is a threat, and it should go ahead and select B cells to form the basis of immune memory and having memory IgEs around, right? So that the next time it's exposed to it, you get a hypersensitivity reaction. And not surprisingly, certain shapes of antigens that we tend to be more allergic to are the ones that look more closely resembling these types of enzymes or proteins secreted by things like parasites, because evolutionarily, right, it makes sense that our immune system is pretty good at identifying these as potential threats. Recall also that a lot of our discussion of allergies has to do with the concept of the hygiene hypothesis, right? Why is it that in modern society, especially in first world developed countries like the United States, we're seeing almost like an epidemic of allergies, right? And a lot of it might be the result of our modern lifestyles, being too clean, being too sterile, having too much access to modern medicines that shorten the length or prevent infection from certain diseases. And as a result, our kids, right, their immune systems as they're developing are not getting the proper education or training or exposure to these naturally expected types of parasitic threats or pathogenic threats like viruses, bacteria, whatever, that Mother Nature expected us to be exposed to as part of training our immune system. And as a result, our immune systems are getting more and more sensitive because they try to calibrate. Our immune systems are constantly calibrating. And if it doesn't detect, normal levels of pathogenic antigens, then it gets more and more sensitive until the point where it starts making mistakes, right? Like it sees the papaya proteins as being a threat, or it sees pollen from a flower or cat dander as being a threat. All right, so this slide just summarizes the cells that are involved in type 1 through 4 hypersensitivities. Again, it's a nice one to kind of talk through and ask yourself, does it make sense? Why are certain cells involved with certain models of hypersensitivity, whereas other cells might not be relevant? All right, chapter 15, we talked about grafts and transplantation. We said that HLA polymorphisms, right, are the main thing that causes tissue rejection in a transplantation. ABO blood type mismatch, of course, is just as important, right? But we usually don't worry about that because being able to properly match ABO blood type is actually a trivial test. I mean, obviously, you hope you don't screw it up, or the clinic doesn't screw it up when they're preparing the patient, you know, for matching with a organ donor. But the HLA matching is a lot harder, right? Because of our polymorphic nature of the HLA. And this illustration, again, was just to remind you, right, that we evolved to have a wide variety of different alleles or genetic sort of makeup of our different HLA proteins that make these MHC platforms that our cells use to present peptides to our immune system. Some of them are highly polymorphic, like HLA-A and B, right? Look at this one here, HLA-B, and there's over 2,500 different cataloged, identified genetic variations or alleles in the human population. Right? And why is that good? Because the different variations you have, or the more variations that are in the population, the better chances you have of having right or correct shape MHCs that can optimally hang on to different peptides from pathogens and present them, right? So in this cartoon, you know, I hope you guys aren't sick of this cartoon, because you can already tell I love this cartoon. I always come back to it because I think it gets the point across very nicely, right? That if you have a pathogenic protein, for example, as a result of some pathogen infecting your body, like a virus, right? And this protein is broken down inside of an infected cell into little peptides, right? So this chicken is broken down into all its little parts. The more different parts of the chicken that your cell can successfully present on its outside, the more potential unique T cells with unique T cell receptors can participate in actually seeing the thing that their T cell receptor is the right shape to see, right? So for example, if this T cell over here, right, is only designed, it has a T cell receptor that's the right shape to detect the chicken head here, right? Then good, right? Because this cell is successful in presenting the chicken head so that T cells can see it, right? But what if, due to the polymorphism here that this particular person's body has, right, they don't have the right shape HLA to present chicken head? So that means your cell might present the wing, the foot, whatever, right? But if you have a T cell that's looking for chicken head, it's not going to see it, right? And so as a result, this particular person's immune system might be at a disadvantage because you're missing out on the ability to actually show certain populations of T cells a particular peptide signal, right? Whereas somebody else who has a different set of polymorphisms and can show all three of these things, the head, the wing, the chicken foot, that means they can activate all three types of T cell receptor that have T cell receptors that recognize all three. So therefore, they would have a stronger, much more quickly acting immune system, right? And that might make all the difference between them, you know, surviving an illness and somebody else not surviving, right? So as a species, it's good to have these variations. But as an individual, I mean, you're just stuck with whatever you're stuck with, right? Whatever mom and dad gave you in terms of your set of polymorphisms, I mean, it is what it is, right? So maybe in some cases, you have a good set of polymorphisms for a particular type of pathogen. In some cases, maybe you're out of luck, right? And you won't do such a good job of having a good immune response for some other type. So recall that when T cells are involved in tissue rejection, this could either be direct or indirect. So direct rejection of foreign tissue is pretty straightforward, right? So here you have a dendritic cell that came from the donor. So this is maybe a dendritic cell that hitched a ride in some transplanted tissue, like a kidney or something like that, right? So this donor cell, it's in the new person's body. So it's like, hey, all right, I'll go back to work, go to the lymph nodes and start presenting stuff. The problem is, right, it's presenting it using the donor's MHC, reflecting the donor's HLA type, right? Whereas the recipient or the patient's body, their CD4 and CD8 cells, right, might just freak out because the shape of the HLA is so different that they just start activating, right? And that can lead to a lot of T cells then attacking the donated tissue. Indirect allorecognition takes place when, let's say, the HLA matching of polymorphisms between donor and patient was pretty good, right? So the HLA genetic variations are pretty closely matched, so you don't get direct rejection of the tissue. But again, over many months and many years, eventually, because the donated tissue, I mean, this is the amazing thing, you think about it, right? If you're donating a kidney from somebody, right, for the rest of the life of that kidney in the patient's body, all of the cells on that kidney, right, reflect the donor's genetic makeup. That's kind of weird, right? It's not like it really becomes, right, the patient's own kidney. Right? It still reflects the genetic makeup of the donor. And so that means the MHC proteins on the surfaces of like the kidney cell, right, will always be the donor's type. And eventually, the recipient's immune system, after repeatedly just being shown stuff like the little yellow dot here, right, which reflects the donor's MHC type, right, eventually over months and years, you're going to wake up and activate enough CD4 cells that just recognize the yellow dot as being something weird, that you're going to end up selecting for B cells that also have IG that recognize the funny foreign donor HLA type, right? And so this will lead to rejection of the tissue. And that's why I told you that even in the best case scenarios, a successful organ transplantation, right, is successful if it can last maybe like 10 years. And after that, 40% of all tissue rejections that have made tissue transplantations that have made it that far, a whole decade of working, right, 40% of them fail after that. Okay. We also learned about graft versus host disease, that situation where some of the transplanted tissue, if there are immune cells from the donor that hitched a ride, they might actually end up freaking out against the tissues of the new body because they're seeing things that they don't like. Okay. So that would be graft versus host disease. Okay, chapter 16, then we moved on to autoimmunity. All right, when we said autoimmunity is very different than immunodeficiency. Immunodeficiency, again, is when there's some component of the immune system that's lacking or missing, right, or has been eliminated. Autoimmunity, right, doesn't necessarily mean that at all. You could have a perfectly healthy, intact, robust immune system, but autoimmunity means one day it makes the terrible mistake of losing self-tolerance training. Right? So remember that much of this has to do with a genetic component. Right? So most autoimmune types of diseases have some sort of genetic risk involved in it. And a lot of that has to do with, again, our HLA proteins that are part of our major histocompatibility complex. Right? So you'll remember that we call them the MHC genes because they are the group of genes that are all located on chromosome 6 in this particular stretch of chromosome 6 where these HLA genes are, right? And so the class 1 HLA genes like A, B, C, E, F, and G, right, are located over here, and they code for the HLA A, B, C, E, F, G proteins that make up the MHC class 1 platforms. Then you have this class 2 region, the HLA-Ds, right? And there's several versions of them like DP, DOA, MB, et cetera, et cetera. I didn't require you to know all of those because it gets very confusing, but I wanted you to understand, right, that these HLA-Ds, right, are all located over here, and they code for the different versions or variations of MHC class 2 platforms, right, that all involve HLA-D. Either DM, O, P, Q, and R, right? So we said that the genetic component of autoimmunity often has to do with whether a person has certain shapes of MHC platforms on their cells that do a better job of presenting certain types of, let's say, peptides that immune systems can make a mistake against. So we said that because these genes are all inherited as a haplotype block, meaning because the genes themselves are physically close to each other on chromosome 6, in that region, they are inherited as a set of polymorphisms that travel together, right, from parent to offspring. And so certain haplotypes or allotypes then have been shown to be more associated with disease risk. So here on the right is just an example. So this particular haplotype block is showing you what versions or alleles of the HLA-A, B, C, and D genes this particular set looks like, right? Remember the whole like nomenclature with the asterisks and the colon, that's just a way of cataloging and keeping track of particular allelic variations here. Right? So if you have this particular set that you inherited, then you are at increased risk of, for example, these types of diseases over here, including things that we talked about, like celiac disease and type 1 diabetes, right? Multiple sclerosis, rheumatoid arthritis, etc. Go back and review these examples that we chose and actually looked at in terms of examples of autoimmune diseases. Right? So recall something like rheumatoid arthritis, right? Depending upon the genetic variation that you have of your HLA-D, RB gene, if you have, so you remember in this cartoon, here's the MHC2 platform. It's made up of an alpha and a beta side to it. If you just look at the beta side, the red part is where it would actually be holding on to the peptide that's going to be displayed on this platform. Now looking down straight from top to bottom, looking down on it, you can see, right, this is the cleft where that peptide would be held. And the red dots here represent different amino acid positions in the beta chain here, or beta side of the MHC platform, that people would have different variations in amino acid identity in. And so over here we said, for example, if you just looked at amino acids position 67, 70, and 71, right, depending upon what amino acid you have in that position, based upon the polymorphism that you inherited, that means your cells may do a better job of presenting a particular peptide or a worse job of presenting a particular peptide. And if that peptide is the one that the immune system makes a mistake against as part of developing rheumatoid arthritis, then you can see your genetic makeup then will contribute to how likely that mistake will be made, and therefore how likely you're at risk of developing rheumatoid arthritis, or conversely, how you are actually protected against developing rheumatoid arthritis. All right, so the last chapter then is cancer in the immune system. We said that absolutely we should appreciate the fact that our immune system plays a huge role in preventing cancer from forming in our cells on a daily basis, right? Because our first line of defense against cancer is that cells that get mutations and begin to transform or start heading down the path to being cancerous, our first line of defense is those affected cells should do the right thing and undergo apoptosis by themselves to take themselves out of the picture. Right? But if the mutations that they got actually interfere with them being able to undergo apoptosis, and those cells stick around and start dividing out of control, then our second line of defense is our immune system. Right? And our immune system has to step in, identify those cells, and say, hey, not so fast. You need to undergo apoptosis. We'll make you undergo apoptosis. Right? And the thing is, this happens every day in our body, right? Not just once or twice, but hundreds of thousands of times over, right? As every day in your body, you have a huge number of cells that get random mutations and start down this process. So our immune system plays a huge role in protecting us against developing cancer. As evidenced by that, we looked at these early experiments where if you take genetically inbred mouse strains that are used as lab sort of animal models, right? And you actually you can see that if you have tumors, cancerous tumors that grow in one genetic background of mouse, like the yellow one, right? Those cells, if implanted in another genetically identical yellow mouse, will also develop into cancer. Whereas if those same cells are injected into a different genetic background mouse, like the blue one, because of the differences in the immune systems, right, the different MHC types, the blue mouse its immune system can fight off the cancer cells. And that's why I said cancer is not contagious in the way that we normally think of things being contagious, like the cold or flu or whatever, right? Because if you get cancer cells from somebody else in your body, your immune system will identify them and destroy them as a foreign invader. We said that transformed cells that are becoming cancerous, they can present all kinds of abnormal peptides. Either, for example, on the left, bottom left here, mutant forms of the peptides reflecting the mutations that the cell has gotten, right? Or things like certain genes that should not be turned on in this particular type of cell, right? So the cell should not be making this red protein, right? And now, but instead it is, and so peptides from the red protein are being expressed. Or the cell is making green protein just like it normally should, but it's lost control and it's making way too much green protein. So any of these three scenarios can be a scenario that the immune system can use to detect that something is wrong with the cell and maybe the cell should be destroyed. We did talk about how certain mutations in cancer cells give them clever abilities to avoid the immune system. So for example, in this cell here, it has mutations causing it to produce high levels of TGF-beta. This particular cytokine attracts Treg cells, right? So you'd have a whole bunch of these orange Treg cells surrounding this cancer cell. And because you have lots of Treg cells there, the Tregs, what do? They inhibit other lymphocytes, like a cytotoxic T cell or this other helper T cell, from properly activating against this cancer cell. And lastly, we discussed therapeutic options of leveraging the immune system to fight against things like cancer. And so one of the hottest areas right now is the development and use of monoclonal antibodies. Because again, right, the point is that a person who develops cancer, the fact that they have cancer, it does not mean that there's necessarily anything wrong with their immune system. It's just their immune system has failed to properly detect that cancer growing inside of them, because those cancer cells had sort of stumbled upon the right set of mutations to make them sort of invisible to the immune system. So if you can wake up the immune system and get it to pay attention to the cancer, right, that is much better and stronger than any kind of synthetic drug or compound that you can give somebody, right? Because Mother Nature already designed us with a super strong immune system to be able to detect cancer cells, right? So if you can just wake it up and encourage it to do its job, right, that is probably the most powerful way of fighting a person's cancer, either through the use of monoclonal antibodies, right, that can either cover the cancer cells and recruit other immune effector cells to destroy them, or as we learned, you can actually tether or link different types of things to antibodies, like cytotoxic drugs or like radioactive isotopes. Right? And then finally, we looked at examples, for example, in the case of fighting prostate cancer, how there are strategies now for actually retraining a man's dendritic cells to preferentially show cancer peptides, so that once those dendritic cells go back to work inside of the man's body again, right, it really emphasizes to all of the T cells that these are the cancer peptides that T cells now need to pay attention to. Okay, so that's the end of our summary. I know it's a lot of stuff, but hopefully if you pace yourself through the next few days, you can get through it. Right? And importantly, I always emphasize, make sure you understand the concepts first. Make sure you understand, right, what we were talking about first before you focus too much on all the little details, right? Because if you focus on the details first, but you don't really get the big picture of how everything connects, I mean, you all know it's hard to really feel like you're learning anything beyond just simply memorizing things, right? But if you understand the concepts and you can piece it together in your head, connecting the dots, right, then putting the details onto it, right, you will be much more successful, because then you are really learning, right, and not just memorizing. All right, so good luck with your studying. If you have any questions, feel free to email me, and I will go ahead and post these slides onto Lalima for you to have this afternoon. Okay? All right, good luck. I will see you again. Well, I won't see you again, but make sure, right, you log into La Lima on Monday at noon to take your test. All right. All right. Thank you.